Go.
Spine canal. Here's the aorta, going across the aortic arch. And just to remind you, this is the brachiocephalic. This is the common carotid. And this is the subclavian. I know the word subclavian doesn't mean a thing to you right now. It will when we look at the CAP tomorrow. Okay. So it's A for aorta, brachiocephalic it's a B, C for common carotid, and S for subclavian. So it's ABCs. Okay. What I wanted to show you on this, it's really weird kind of, there it is. If I open this, oh, what is this? There's the inferior vena cava, and this right next to it. Here it is, okay? This is the coronary sinus. Both the opening and the vessel are called coronary sinus. You won't recognize it necessarily though if we turn to the back. The arteries circle the heart, the veins do too, and here coming back, this pouch, this enlargement, is the coronary sinus again. It's going to enter in under my finger. The hole that you saw just a second ago, which we call coronary sinus, is attached right here. So look on the back of the heart. There it is. For the coronary sinus. Done with that. Now we're going to do an easy one. We'll do the ear next. Okay. I would appreciate some help, somebody to try to aim it. That's much better. Okay. Thank you. Turn it just a little bit. No, at the base. It'll fall. That's good. Okay. This part of the ear, which has now taken on the role of ringlet depository, okay, all the way around. This is the pinna. This is the pinna. If you study otology, the ear, you get a name for every little fold. Okay. But we're just worried about this one, pinna. This is the auditory canal. Now, let me see what I have. Auditory canal, okay. The auditory canal is where people put Q-tips. If you ever read the Q-tip box, it says, don't do that. But if you listen to the Q-tip people and don't do it, you'll put them out of business. They know you're gonna do it. Okay, so, this is the auditory canal. It continues back until you get to this structure here. This is the tympanic membrane, or simply called the tympanum. I don't know how these ears get broken. You take this one. Okay. This one's better. Here's the tympanum. I'm going to take it out and show you that two of the ossicles are attached to it. The one attached directly to the tympanum is the malleus. This one that looks like a tooth, it's upside down, but it's a tooth-like structure. This is the incus. We've learned these before, of course. The stapes isn't attached in the model. It's up here. Looks like a stirrup. We'll come back to that in just a second. Behind the tympanum, you have another tube. This is the eustachian tube. Now I want you to write, please, for yourself, the function of the eustachian tube. We'll be writing on this page quite a bit. The eustachian tube helps equalize pressure on both sides of the tympanum. You have pressure outdoors pushing against the tympanic membrane, and if it pushes too hard, it misshapes it, and your hearing gets fuzzy. So you open your mouth, take a swallow, chew gum, or whatever it takes, and then you open up the eustachian tube. The eustachian tube allows the air pressure to equalize by sending air on the other side of the tympanum. You ever go up to the Palm Springs Tramway? Anybody? Going up's not bad. Coming down, your ears don't pop fast enough. You know, I've seen people screaming on that little ride. Okay, up above here, there's a good shot of the stapes right in here. And the stapes attaches to the vestibule. See this area here, the chamber. Fine vestibule input contains semicircular fluid. Contains semicircular, that's one word, fluid. That same semicircular fluid moves into these semicircular canals. And the semicircular canals are for balance. Please write that down. Balance and equilibrium. you can cross off the word ampulla. This structure over here is the cochlea. The cochlea has within it the sensory cells for sound. Each sound or each sensory cell responding to different pitches. Here's the cochlea. On most of the models, and you can piece a whole model together. There's a couple up there that are perfect, actually. On most of the models, though, the cochlea is split in two. I just took half of it off. It doesn't matter whether you see it that way or you see it this way. It's still the cochlea. Okay. Anything else on there? Else at the stage? Oh yeah. Vestibulocochlear nerve. It's back here. There are two nerves. There's a nerve here, big one, and a smaller one. This is the cochlear nerve going to the cochlea, and this is the vestibular nerve headed for the vestibule. Together they're called the vestibulocochlear nerve or cranial nerve 8. You'll need to know by that. Cranial nerve 8. The eye. We have many models of the eye over there. If yours is not perfect, just get another one, because there's plenty of perfect ones. There's only one, and that's why I chose this one, though, that has the cornea present. Okay. We are going to do a sheep eye. It's pretty easy. I'm going to save that for first thing tomorrow. This is the cornea. The white connective tissue, the whites of the eye. This is the sclera. It's a very protective and containing layer. It's the thickest layer of the eye. Ever known anybody to have an injection into the eyeball?
Yes, for macular degeneration.
Well, it can be for that. It can also be for any disruption of the retina itself or inflammation of the optic nerve. Yeah. Sounds horrible. Have somebody poke a needle into your eye. But not quite as bad as it sounds. Okay. You recognize the muscles. Okay. This area in here, in front of the iris, this is the iris or iris diaphragm. The iris diaphragm is the colored portion of your eye. Iris can increase or decrease the size of the pupil. Please write it down. Changes pupil size. That means it lets more light in or excludes more light. You have pupil on there as well. There is no structure that corresponds to the word pupil. Pupil is an idea. Pupil is the opening through the diaphragm. So let's put that down. Pupil opening through the diaphragm, or opening left by the diaphragm. When we have a door to the room, you walk into it, you know it. But if the door is open, it's a doorway, and you can't hang anything on that doorway. The area in front here is the anterior chamber. Can you find it?
Anterior
Anterior
segment.
segment. Okay, same thing. Actually, there's a slight difference. We'll go with the anterior segment. The anterior segment contains a fluid. It's called the aqueous humor. We need that to push the cornea away and keep proper spacing. The anterior segment contains aqueous humor. Now I'm going to take the top off. The first layer was the sclera. This darker layer here is the choroid coat. Choroid coat. If we wanted to see it on a living or dead preserved specimen, we need to cut into the eye. The model is able to do it this way. Here's the choroid coat. And the function of the choroid coat is to absorb excess light. Write it and I'll explain it. Once light goes in through your lens, it reaches the retina, where it strikes receptor cells. But it goes right through the retina too, and goes to behind the retina, and back there is the choroid coat. The choroid coat absorbs the light that came in. If that weren't happening, the light would bounce around inside your eyeball many, many times, and it'd be very difficult to, focus on anything. Okay, so this is the choroid coat. It absorbs excess light. At the back, this is the optic nerve. Take off the lid here, see where I can get it in focus, that's it. Now, you don't have this structure. You don't have this plastic structure in your eye. I'm going to take it out in just a second. It's to indicate where the posterior segment is. You have that? The posterior segment, much larger, and it needs to stay inflated. It needs to have pressure enough that it pushes the retina against the wall and keeps it in place. You don't want too much pressure though, you don't want glaucoma. The posterior segment contains vitreous humor. Aqueous humor in the first segment is watery. Vitreous humor is like jello. Not all the time, but depends on the preservation, but usually it's like jello. So the lens is here, I take away the posterior segment, the piece there representing it, the lens will leave too with me. So the crystalline lens is to focus the light. Now I'll take this out. Take the lens out. What is this, orange? What color is this?
Orange.
I got one?
Yeah.
Okay. Okay. This orange material is the retina. And you do have the retinal arteries and veins in there. The retina perceives the light, and the light continues on through the retina and ends up back here at the choroid coat again, which the job of the choroid coat was to absorb that extra light. There, I got it. Okay. Now, the retina comes together back here. We said that this was the optic nerve back here. But here, this is not the optic nerve. This is the optic disc right here. Is it yellow, I think? Yellow circle. The optic disc does not perceive light, so you don't see anything with the optic disc. It's where the retina and the optic nerve are exiting. It's a blind spot that you have in each eye. On your model, look for this little piece right here. What color is that? I have no idea. Purple? Okay. Right there. That little spot is the fovea centralis. It's where light would focus if you had perfect vision. If the shape of your eye was perfect, you weren't nearsighted, you weren't farsighted. Now, if you wear glasses to correct a condition, the glasses are meant to focus the light on the fovea centralis. Turning it around, the lens went in here. It's gone now. But you can see attached to the lens is white, looks like a tire. Okay, the white portion. That's the ciliary body. The ciliary body attached to the lens can pull on the lens and make it change its shape. Especially when you're younger. You hit 40 and the lens doesn't want to give very much. And the ciliary body can't help out. The ciliary body is important in going from distant viewing to close-up viewing. Or the other way around, you're reading a book, you look up and see what's coming. You have to change. You have to change, and the lens will change. If you find that that change is slow, inconvenient, not working, people tend to head right for reading glasses first. You know, cheap ones at the drugstore. That'll be the end of it. You start doing that, and you will have to use them forever. Well, there's worse fates. I know people who buy expensive glasses. It's like a foot fetish from shoes. They've got to have these glasses. Okay, now, you see the area where the cove is now? No little white lines or anything. Okay. It fans out even farther. That is the ora serrata. Some books will tell you it has no function, but I just disagree with that. When light comes in, we know if it hits the retina, it'll go behind it to the choroid coat and be absorbed. But when light comes in at a really tight angle, coming in from the side here, goes through, and on your model, when you get to it, you can see the ora serrata. And that's what it's hitting. The ora serrata absorbs extra light. It does not have a muscular function like the ciliary body. It's there to absorb light at the front of the eye. And where it ends, the retina begins. This is a pretty good one, so I'll leave it out here if anybody wants to use it. And now, the brain. I took two brains. It's not going to be hard. I took two brains so that I don't have to cut into both. Oh, somebody cut into this one. How horrible. Let's see what it looks like. Oh no, it's good. It's good. It's good. Okay. Start with this one. I'm holding it by the spinal cord. I'm trying to get some focus on it, but I can't do it myself. This gland is the pituitary gland. Pituitary gland. We'll talk about the functions of that one separately. We'll talk about all of the hormones that it secretes. Here and here. You see both of them here. These are olfactory bulbs. Also, you can call them olfactory lobes. On the underside, here's the spinal cord, and then it swells right here. This area is the medulla or medulla oblongata. Higher up, spinal cord, medulla, and then these two bulb-like structures. 1, 2. Those are the pons. P-O-N-S.
Which one?
Pons, medulla, and I'm holding it by the spinal cord. The next thing you would do, you could either use the same brain, you're going to bisect it longitudinally, or you can use a different one because you want to preserve the pituitary gland. The pituitary gland often falls off and you cut it, as does this entire section, but it hung on in this case. When the brain is cut longitudinally, the front here, all of this material is the cerebrum, area of higher thought. All of these will be given to you on a filled out sheet. You don't need to write for yourself unless you wish. Here's the cerebrum. Right below it, there's an area here. I'm going to make a hole in it because the hole does exist. This hole is the lateral ventricle. There's one on the other side. The ventricle's function is to contain cerebral spinal fluid, to nourish the brain and also give it a little bit of shock resistance by having the fluid inside. So notice that around the lateral ventricle, there's this tissue, rather thick. This tissue is called the corpus callosum, which means the tough body. This rounded structure, I call it a baby scallop. The rounded structure is the thalamus. Thalamus.
Can you point to it again?
What?
Can you point to it again?
Here.
Oh.
You know what I'm going to do? I'm going to turn it around to the other side. Okay. Here's the thalamus. Here's the corpus callosum. I think it's a little bit better over here. This structure here is the corpora quadragemina. Some people think it looks like a pith helmet or a hat. Right next to it, they managed to leave this in cutting it. Okay. This is the pineal body that secretes melatonin for sleep control. They managed, in fact, to cut this so luckily that it sliced the pineal body exactly in half and left half on each side. Usually, that doesn't happen. Usually you'll get it on one side and can't find it on the other side. Back to the thalamus, and now go under the thalamus, right in here, just the tissue under it. There's nothing special about it. It doesn't look special, but it's the hypothalamus. The hypothalamus is the seat of the autonomic nervous system. The structure back here and on the other side is the cerebellum. It controls more primitive things like breathing. It has the outline inside of it of a tree or a shrub. That's called the arbor vitae. The thalamus is the whole thing, but the arbor vitae, which means living tree, is the pattern. You will be asked to distinguish between those two terms, cerebellum and arbor vitae, but it'll be clear because we'll say, name the pattern. Another ventricle is found right here, right at the bottom of the tree. You see a little split there. And you can see possibly the little split on the other side as well. Now, this side's good enough. This is the fourth ventricle. That lateral ventricle and the lateral ventricle on the other side are two separate chambers. The fourth ventricle is a single chamber. There's a third ventricle as well. You can't see that. The lateral ventricles used to be called numbers 1 and 2, but they went with lateral. So anyway, there's 4 chambers in the brain, all of which contain cerebral spinal fluid and protect against shock. Now I'm going to ask you to number the brain parts, okay, on your sheet. 1, 2, 3, straight down. You should end up with 15.
Yes.
Okay. Tell me number 1, please.
Cerebrum.
Cerebrum?
Yes.
Okay. Don't worry that you can't see it here. You're going to see it over on the specimen. 2?
Cerebellum.
Okay, now I can poke it on the outside or the inside. I'll put it here. 3, please.
Arbor vitae.
Arbor vitae. So there's 3. 4.
Lateral ventricle.
5. Corpus callosum. Got it? 6. Okay, for that I'm going to go to the other one. It's off screen, it's okay. 7. I'm not hearing it, it's mumbling. Okay. There's 7. 8. Pineal body is right next to it. Ready for 9. 10,
One.
pituitary gland, I'll do it here. 11,
Hypothalamus.
hypothalamus. That needs to be raised. Twelve,
Fourth ventricle.
thirteen,
Medulla oblongata.
14, does that be Hans, right?
Yes.
I'm gonna have to lift that up to see it, but that's a beautiful one. And then 15 would be the spinal cord, right?
Yes.
And so that's that. Your job's to review what we just did, okay, and hopefully get down as much as we can before you go. Okay.